So one of the issues with current drug release methods is that we more or less just kind of dump and pour the drug into the bloodstream and hope it gets where it needs to go. So a couple of issues that come from that is that tend to have really big side effects because we're putting a large amount of drug in the body, in the bloodstream, and it goes where it's not supposed to, and you end up with bad effects. Similarly, you don't have it's not a very efficient release method because you have to use a lot of drug just to get a small effect, since the target site is such a small amount of the bloodstream of the body. So a couple of ways that we can improve upon that is to use hydrogels. Specifically, we've looked at using stimuli responsive hydrogels. So the advantage of using hydrogels is that they're much better at getting to the target site and staying there, and then using sustained releases that's a little more continuous and more efficient and doesn't use as much drug as just the dump and pour method pretty much does. So specifically, stimuli responsive hydrogels are ones in which they receive an environmental cue and then undergo some significant change, and we can use those to control the drug release process. So specifically, our research group here at Ziegler Lab has been using polynipam hydrogels, which here are a couple of them dissolved in solution. Polynipam at around 32 degrees Celsius transitions from a hydrophilic dominant state, so it really likes water, to one that doesn't like water, and it pushes all its water out. So if you were to trap a drug inside of polynipam and then have it transition from its water liking stage to its water hating phase, it would push the drug out with it. So that's would be a much more efficient method because you can control when to release it. But of course the issue is that the body regulates temperature very well, so you have to be careful about what temperatures you use. So what we do is we adjust the formulation of our hydrogel in order to get the temperature right above body temperature, and then we actually use a nanoparticle called single-walled carbon nanotubes, which can absorb certain wavelengths of light and then heat up. So what we're essentially making here is two such samples. Essentially what these do is the carbon nanotubes act as a remote trigger for the heat response, and then we can use that to remotely outside the body trigger drug release inside the body, which would make it so that on demand you can release a drug. So this process and this project has really involved a lot of chemistry and nanoscience. I've learned a lot from those. And of course, one of the challenging things is that in the back of our minds, we always have to be thinking about biology and what applications these would lead to, because eventually we want to put this inside the body. So it's really expanded my knowledge of science and helped me kind of understand more things. And specifically, one of the things that I learned was more or less that there's not one answer to the problem. A lot of times there are multiple. I stopped asking people the way they would do it to ask what their answer was, because I realized that there isn't just one answer. Instead, I started asking people more or less the background of things and kind of their experiences, so that then I could take some of their knowledge and experience and kind of form my own answer to problems. And another thing I learned that has been really helpful to me is to be able to take knowledge that I've learned in classes and just reading academic papers, and then taking those and comparing those to observations we see in the lab when we mix different things and do different things to our hydrogels, and trying to come up with a logical response to those tangible problems.